First, I'm going to talk about KRAS and KRAS lung cancers and diagnostic testing. So, among all individuals who have lung cancer, about a quarter of these individuals have what's known as KRAS mutant non-small cell lung cancer. So, lung cancer itself is divided into non-small cell and small cell lung cancer, and KRAS is a form of non-small cell lung cancer. Shown on the right-hand side, not only is KRAS mutated in these cancers, but it has a different mutation in different KRAS mutant lung cancers. The pie charts show the distribution of the actual mutation itself. And these mutations change just a single amino acid in the KRAS protein. The most common mutation is on position 2 of the amino acid known as KRAS G12C, which changes a glycine at position 12 into a cysteine. But you can see here that KRAS G12, G13, and Q61 are all forms of KRAS mutant lung cancer. So why does this matter, and what is KRAS? So KRAS, which stands for Kirsten rat associated protein, is actually a normal protein that's in all cells. And you can think of it acting inside the cell as almost like a light switch. And when it is turned on, which is when it is switching from the GDP bound off state to the GTP bound on state, it signals the cell to grow and divide. On the right-hand side, you can see that the KRAS protein positions 12, 13, and 61 are actually quite close to the GDP pocket. And just that minor switch from a G to a C at position 12, for example, changes KRAS to favor the GTP bound on state. And so in this scenario shown on the left-hand side, inside these cancer cells only, KRAS is enriched in this on state within cells, and then it signals to a variety of proteins, including some of the ones here, which we're developing new treatments for, and tells the cells to grow and divide. And ultimately, cells that grow and divide are what cancer cells are. And so this is what ultimately drives KRAS-driven lung cancer. A history of KRAS-driven lung cancer is that we know actually, and we've known for a very long time, that KRAS is a mutation in KRAS is necessary and sufficient to lead to cancer cell growth. This actually derives from work starting in the 1960s, and KRAS, among other genes that are mutated, led to the development of the oncogene theory in 1969. Finally, in 1982, we were able to define that KRAS itself is an oncogene, and so it causes just this mutation alone causes cells to grow and divide when they're not supposed to. And in the 80s, we identified different carcinogens that led to mutations that led to proteins like KRAS being mutated, leading to cancer. Then it took several decades to really identify how we could shut off this signal. And it's because targeting KRAS is a challenging problem. And so it really took many decades of persistent scientific effort until 2023, when there was a breakthrough from Keyvan Shokat lab and colleagues, where they found that the mutant form of KRAS had a specific pocket in the off or GDP bound state that you could introduce a small molecule inhibitor to take to shut off that signal. And so ultimately, this has led to the first FDA approval. It was an accelerated approval given the initial promising results of the first KRAS G12C inhibitor in lung cancer known as sotorasib. And since this breakthrough, there's been really quite renewed interest in targeting KRAS-driven lung cancer. And since then, there are multiple different ways of targeting KRAS, including inhibitors similar to sotorasib, those that are binding other locations, immune approaches to these lung cancers. And so many KRAS trials are actually active and enrolling currently in 2024 in non-small cell lung cancer. So, what does this mean for someone who is presenting to the clinic with a new diagnosis of lung cancer? So, because we have all these promising potential clinical trials and different treatments depending on the subtype of lung cancer in this era of precision oncology, every single diagnosis of lung cancer really needs not only the pathologists to look at it, but also understanding the mutations and also the stage, which is where is the cancer. And so, illustrated here is someone who presents with a new diagnosis of this type of lung cancer. And the first thing to pursue is diagnosis and staging. And so, in the medical oncology clinic, what we're asking ourselves are what kind of cancer is this? What kind of subtype? What are the mutations associated with it? And what organs are affected at baseline? This informs us about what are the personalized treatments that we can think about that could potentially eradicate the cancer, and if eradication is not possible, control and or improve the quality of life. And so this is the key question that oncologists are always asking themselves. Additionally, other considerations include what are the current cancer-related symptoms, and can some of them be addressed right away with other treatments? And what other medical conditions does this individual have that really should be attended to because they will help the person get control of their cancer better and or improve their quality of life. And so, as a part of this, that first clinic visit not only includes a history of everything that has happened related to the cancer, a physical exam, laboratory tests, scans, and imaging to find out the stage of the cancer. But a key component of this, which actually does take weeks of time, is the initial pathology assessment, which is sampling the cancer and actually figuring out what are the characteristics of this cancer. And so for KRAS driven lung cancer, generally the histology or the appearance under the microscope is that it is a form of non small cell lung cancer. The most common subtype for KRAS driven lung cancer is lung adenocarcinoma, although we do see other subtypes. Additionally, in order to identify the KRAS mutation itself, there is DNA sequencing that needs to be performed. And so this is done by a molecular pathologist, so someone even further trained in pathology who actually goes through all the DNA within the cells on the molecular level that are sequenced and decides whether these mutations in KRAS are present or not in the cancer cells. There's two ways to do this. One of the ways is directly from sampling the tumor, and the other is looking in the bloodstream. Once we have all of this information, that is how we develop personalized treatments for lung cancer. And then we start the treatment, and subsequent clinic visits include history, physical exam, these tests, scans, and imaging. And that helps us decide if this treatment is working or if we need to change course. Among the decision making, there is a chance that the doctor will ask for a subsequent pathology assessment. And so a repeat biopsy may need to be done. And once again, this would be to look at the histology again and also identify are the same mutations present or are new mutations that have developed. And this does actually guide treatment.